Well, thank you for a wonderful talk, and thanks to your mother as well, I should say. On that note, with regard to the sequencing being so close to becoming commonplace, how prepared are countries, healthcare systems, and insurance companies for what could be a very rapid rush toward an onus on preventative care? And what does that mean towards systems that are very heavily focused on treatment as opposed to preventative care?
Yeah, another really complicated issue. So first of all, in principle, you're not supposed to be discriminated for health, for medical care, and for employment based on your genetic information. But you can be discriminated for life insurance and for long-term disability. So there are issues there. Insurance companies, for the moment, they're a little bit in the wait and see. I haven't heard anybody getting, even me, nobody's had, of course, I'm tenured professor at Stanford, that's probably a little unique. But I haven't heard anybody getting truly genetically discriminated against because of their genome or their genetic information. I have seen people benefit through the undiagnosed disease or the cancer, and that stuff, a certain amount of that is being reimbursable. It depends on the insurance company, it depends on the situation. So some of that is falling in, is getting handled. Our system is a bit screwed up for handling this properly, quite frankly. If you think about it, the insurance companies should be paying us to get our genome sequenced because you would use that information to better manage your health, right? A person who gets their genome sequenced is probably going to be healthier than a person who doesn't because they're using that information in a useful fashion. The problem is, if you get your genome sequenced, well, 2 years later, you may have a job with somebody else. That's a different insurer. And your insurer doesn't track your genome. And so there needs to be a different incentive system. The whole thing is just not right. It's harder to do in the US kind of system. The European system is a much better model that could actually handle this better.
And that's what I was alluding to, really, is places like Britain where you've got the GPRD database, which could be basically the template to expound out to a universal database, could actually put those countries at more of an advantage for trillions in efficiency savings in a blue sky world, as opposed to here, where things are very much focused on the treatment as opposed to preventative care.
Yeah, and that's just the wrong approach. We've got to be at the predict and prevent and catch early and not at the catch it when somebody has the symptoms. It's just too expensive, too hard. And that's where you'll see cost savings too, if we can shift that. And you can argue, well, that just pushes people to later in life. It just shifts costs back 20 years. And that, again, that's a very separate economic argument. But anyway, I do agree philosophically with what you said. Yeah.
Okay, 2 questions. One, on one of the pictures that you showed on one of the lists, it had Alzheimer. Is that connected to genes?
Oh, yeah, sure. There are loci that are high risk for Alzheimer's. And it's the number one thing people worry about when they get genotyped either by one of these companies or get their genome sequencers. It's called an
So it is connected?
allele. Yeah, there's one that's very highly predictive. As a male, you have about a 17% chance of getting Alzheimer's without it. And you have about a 25% chance you have 1 copy change, and about a 60% chance you have 2 copies change. Okay. So that one, 2 copies, meaning both copies are changed, is, again, it's not 100%, but it's very predictive. And that's why you need to be properly, nobody should get their genome sequenced without genetic counseling. I forgot to say that. I do think that's very important. Because you may learn you're high risk for Parkinson's. That is not a curable disease. No. Okay? Now, you may have an inkling from your family history, but it doesn't guarantee it, just like the person with the BRCA mutation.
And now, the other thing is, what I wanted to ask, if someone has a cancer, like prostate cancer, and it's not taken care, it's like spreads like a disease in the whole body. Because we have a friend that had prostate cancer, and then he got cancer in all his bones. So what does it do? It damages other...
Well, see, cancer is really cells growing uncontrollably. And once they leave the site of origin, that's called metastatic, they've moved out, and that's when it's a huge problem. Because you can't cut it out anymore. It's moved to many locations. And it's very difficult to cure anyone. That's called stage 4.
Yeah, but
it's very difficult to control anyone who's in that. And so your bet, but, well, go ahead, what were you gonna?
What I meant is, so does it keep on damaging genes? Or it's...
Well, yeah, that is a problem too. The genes will keep mutating. So there are probably some initiator genes. It's thought that minimally you need 5 genetic changes to get cancer to get started, but it could keep going and keep getting different changes. And so that, and then at the end, the cancer is very heterogeneous. And so that's why one drug may not solve. You may knock down 90% of it, but when you have millions of cancer cells around your body, that 10% can come back. And that's why people aren't typically cured with any of these drugs. In a best case scenario, they may be managed. There's a famous case with CML where it's almost all, that's a rare case where it's almost always a particular mutation. And there's basically a drug that came out that targets that. And it really sent, almost 100% of the time, sent all these patients into remission. Not 100%, but nearly 100%. And everybody thought this is it, they're cured. But actually 5 years later, back would come these recurrent changes because there were some cells there that actually got around or they got drug resistant. So it is a complicated thing, and that, I would argue that's why we're going to need 2 drugs. This is where the field is heading towards, but we're not there yet. Should probably keep switching here.
From what I've read about epigenetics, DNA methylation influences how genes are expressed. So I'm wondering, does DNA sequencing show anything about methylation?
Yeah, that's a separate assay. That's one of the assays we're now incorporating. I kind of got behind, so I glossed over that at the end. DNA methylation, it's modifications of your DNA, and it's thought to reflect some of the environmental changes you're exposed to. And so, as an example, we're very interested in seeing what happened during that viral infection. Did my DNA get methylated and change? That's one thing we're looking at right now. We've actually done we've gotten the data, but we haven't finished the analysis on it yet. So I actually think it's going to be a separate assay, a separate analysis. Unfortunately, it's about 3 times more expensive than just getting your genome sequenced. But I think in the end, you'll be getting both your DNA sequence and your methylated DNA sequence. And the complication out of that is your methylation changes with the different tissues in your body. So we'd have to do several and figure this out. So the bottom line is very early days for DNA modifications, although I think it's going to be very, very important.
So do any of the commercially available genetic sequencing services look at methylation?
Not really, because it's too early, although you will see it coming down the road. People are now just starting to see methylation and how it affects cancer because it's such a high impact area. And that's just starting to be interpreted scientifically. So it's not yet, none of that's available clinically.
All right,
and
thank you.
Even if you have cancer, you won't get a methyl done because people don't know how to deal with it yet. Okay.
All right, so my question's actually on data. You've mentioned you have data, you have 1,000 studies or so. There's 23andMe, there's Pathway Genomics, there's a few other groups. Are you combining your data so you have a larger database to work from?
Well, this is a complicated question. My data is pretty unique, but we do make it publicly available. You can download it and play with it. So there's, at least that's the first 50 terabytes you can play with. And other groups have, and they call me up when they learn something. And so if you learn something, I'd love to hear about it. There will be, at this level of undiagnosed diseases, there's a lot of this going on. And what hurts us is we just don't have enough to compare. Like I was saying, when you have a person who has a change in one gene, we need to make a match with someone who has that same change and has a very similar disease. And that's the change that's responsible for that. And so there's a lot of discussion. Some grants are just getting funded by the National Institutes of Health to create these databases that will have at least that kind of information and these clinically interesting variants. So people can make these kind of matches.
Will all the companies have to pool their data in so you have all of the data?
No,
to
that's
go
all
from,
done
or-
through the public effort. So places like universities like to share their information, but it's complicated because we can't just, I'm willing to put my sequence out there, but that doesn't mean you should be willing to put your sequence out there, right? You may not want to share your information because you may be worried about the privacy issues. This is where the counseling comes in. And if you're not worried for you, you may worry for your kids. Again, that's legitimate, but you may want to share it. So there's a lot of people who have shared their information, by the way. So it's a complicated issue. in those circumstances, and by the way, just putting the DNA out there without names doesn't guarantee it's anonymous. You may have heard about this. That is, you can put this out there and there's a chance people can figure out who you are. Like for now, people in my family will know if they get their genome sequence and put it out there, someone will definitely make a connection between them and me. Doesn't mean they'll know whether they're a sibling of me. It doesn't mean they'll know which sibling it is, but you can see that you can start making these matches. And so you can't guarantee any of this is anonymous when it's put out there. But I'm a believer in sharing the information because I think as a community we'll be better if we do the scientific. But in cases where people are worried, they're actually private databases with restricted access, so scientists can get at it and clinicians and such, but then the average person can't get at it. I don't know what happens if ultimately those get hacked and stuff, which probably will happen, but I guess we'll deal with that. That's a risk that you deal with, I guess, every time you set up a bank account too, so.
Okay.
Yeah, oh, sorry.
Yeah, thank you for a fascinating talk. As you pointed out, cancer is a highly individualized and unique disease. Potentially, there may be some similarities in cancer clusters, and I was wondering: is there any work on studying genomic variation in cancer clusters? And if so, has anything been gleaned?
Yeah, you mean common kinds of genes that are mutated and things?
Or epigenomic changes, or,
Yeah.
or transcript?
On the gene changes, there's a huge amount of effort to figure this out, and there's clearly a lot of mutations are falling in 12 major biochemical pathways, and so that's happening, but although there are some exceptions. And I wouldn't say we know how many other pathways are out there either, but there's 12 major pathways that people have identified. And for the transcriptome, for example, breast cancer, you can now classify into 5 different categories based on the transcript profiles, and it actually matches up with the methylation profiles as well. So that's coming out that you can start subclassifying. There are now groups, this isn't published, who are actually looking at the proteins and can classify things even further. Again, by adding more information, we can classify this further and further. So that is the goal, to ultimately classify this by the different types, and that should help us decide what treatments to use. Even if you're not mutated for the EGF receptor gene, being up for that pathway may suggest you should use a drug that targets that, right? So targeting a pathway can still be important, even if it's not the one gene itself. It might be something else in the pathway that's wrong. And so this is the kind of information that will be useful as we stratify patients. We should then use the drugs to hit those classifications. Now, some people say, Mike, if you take this to extreme, every person will fall in their own category, and that is true at some level. But hopefully from the commonalities, the drugs we have now will treat a large number of the patients in some useful fashion.
So I guess I was curious in regard to cancer clusters, whether there might be a dominant triggering pathway that is associated with a cluster, or whether any of that's been studied.
Well, yeah, again, they often affect several pathways. Ovarian cancer, people reported 5 different pathways that show up more often than not in ovarian cancer. So there is that. There are other cases where people have used the information to predict prognosis, not necessarily origin. That is, certain kinds of changes are nastier, and you should use more aggressive treatments. That's why people want to be able to do prognostic predictions, because if something is predicted to be more aggressive, then the goal would be to use nastier drugs against those. So it's being used in different classifications. One is deciding the treatment. The other is based on the prognostics. And ultimately, people use this information. This is going to come out too, for following recurrence of cancer. In the future, you'll be following recurrence of cancer by blood tests, looking for DNA in the blood, not by going back to the sample, because a certain amount of cancer DNA shows up in the blood. So that's how things will appear. I know that's not exactly, sorry, I got migrate a little bit off your question there.
Yeah, and maybe this isn't known, but I mean, if you have a cluster of cancers that's maybe associated with environmental exposure, that might be associated with a dominant pathway that might gain more information.
There's some level of that. Smoking's the classic case. There's a lot of lung carcinomas that are associated with smoking. Mind you, there's a lot that's not. And it's unfortunate because people often get lung cancer. Everybody assumes they're smokers, but that's actually not always true, especially in today's day and age. But anyway, there are certain kinds of changes associated with lung cancer, both at the DNA level, the kinds of changes that occur, the kinds of mutations that occur, and the pathways that occur. It's not an absolute, it's a propensity sort of thing. So it does sort of fit what you're saying. Yeah, and how much of that occurs in all the different cancers, I don't know. Oh, she's moving us along, sorry.
You said that there are some problem areas on the genome that were more difficult to sequence than others. What areas are those and why are they difficult?
Yeah, one of those areas is, for example, the centromeres. We have a very simple
yeah.
sequence and it's repeated 1 million times. So you don't know whether it's 1 million or 999,991, and if there's simple little changes in there, where they sit, which we know there are. So that part hasn't been, and so people say, well, that's no big deal because who cares? And that's probably partly true. But there are other regions of the genome which are tough. They're like 100 kilobases, meaning 100,000 bases long, and they're duplicated several times, and some of them have medically interesting genes in them. It's very difficult, just technically, to tell the way we do it. You can't read through this whole region. So it's hard to tell just how many copies there are sometimes and which ones are changing, which ones aren't. So these regions that are duplicated, they get complicated. They're just technical challenges, and I'd have to sit down to describe it with you further. But this is the kind of problems that still exist. So I think when I last looked, there were about 200 gaps in the genome that existed that weren't yet filled. I'm sure some of them have now been filled, but we're not down to zero.
Thank you.
Sure. Yeah.
I have a question about that diabetes trigger. If the virus is causing this, does that actually change the DNA or RNA as part of the process of triggering the diabetes? Because my thought is if it does, then maybe you end up with something which is not exactly what you had when you made the measurement before the trigger.
Yeah. So we're looking at the DNA now. I don't think there are going to be major changes in the DNA. We've done some analysis to say the DNA itself at the sequence level. But the methylation, this modification, is something we're looking at hard. And I know there are some changes because we've looked at that. And we're still trying to figure out exactly how many there are. It's not a trivial analysis, but we think there have been some changes. What I'm not sure is just how many and such. So we're sorting this out. So the answer is there may be. And this may be how environment works. It actually leaves an impression, a modification on your DNA.
Right. Yeah.
It won't change the actually. And that may be important down the road. And minimally, I know it changes your transcripts. But mind you, the transcripts do turn over. So we don't know how permanent that is as an issue or not. But I think the bigger issue is how they're actually affecting the modification of DNA. And it's a great question. We don't know the answer. There are definitely people who have shown that cancer is modified through methylation, obviously, and so that does have an effect. So I think it will be important. I think in the end, getting your genome is just step one.
Sure.
Getting your methylome is going to be probably several methylomes. By the way, I think we'll all have stem cells associated with this too, these induced pluripotent cells. That's a whole separate topic.
Well,
we do for me, of course.
In this sort of sequencing from the DNA down to the final proteins, are there other steps like the RNA or something like that that are more affected by environmental things?
Well, I don't think we know the answer to that,
Okay.
whether things are more. There's a lot of modifications on proteins, phosphorylation in particular, that's very heavily affected by environmental stimuli.
Right.
So I think that's going to be a major area where you see environmental impact. Those tend to be transient. But how many of those lead to more permanent changes, say, through the DNA, we don't know yet. That will be it's a very interesting area to pursue.
Okay.
a
Okay.
great
Not yet.
Yeah, not yet.
Thanks.
That's right.
Last
talk.
question,
Yeah.
and I think it's a simple one. How accurate are they sequencing? If you take a sample from a person and give it to 2 labs, will you get exactly the same
It's
result?
a very interesting point. And this is something our lab, and I forgot to mention, but I actually am conflicted. You can disregard everything I said. I've founded a genome interpretation company called Personalis. We, at the company in our lab, spend a lot of time thinking about this. But I'll show you the issue that we came up with right off the bat, which is we sequenced it with 2 different technologies. This is my genome. This was done a few years ago, mind you, but if you do it again now, we sequenced with 2 different technologies, Illumina and Complete Genomics. And yes, 89% of it matches up, but there's quite a bit. This is just for those single base changes. That tells you there's a lot of this is missing information. It's not that some of it, there are differences called, but often one technology caused something and the other doesn't. And they just didn't make a call there on your DNA. So there's a lot, that, again, you might say, well, yeah, 11%, who cares? Well, when it's your genome, you care, because there's a lot of medically interesting information in that. And what's even more interesting is recently, the company Personalis actually sequenced the exact same DNA twice, fairly deeply, 60x, meaning 60-fold coverage on average, and still saw 2% differences with each one. So we still have more to do to improve the technology. Anything you find, you go back and validate by other methods. I didn't get into the details, but if you see a variant you think might be disease-causing, like that BRCA variant I mentioned earlier, we go back and validate with another technology to show that it's really there.
Do you
So
think
this is an issue.
5 or 10 years from now it'll be perfect and therefore there's a probability to
Perfect is a
wait?
tough, yeah, word. I agree with the group up here, but
I
yeah.
use that.
But I think people are now realizing, partly because of us showing that there is a lot of issue. The biggest issue is that region's not getting covered properly. And we're now making people aware of this, and so companies are spending a lot more time on the accuracy and coverage issue. So I think it will get much better just in a few years. The complicated regions are these duplicated regions where there are subtle changes. It gets very complicated informatically to sort that out. And they'll take longer to solve. Okay,
Thank
but
you very
others
much.
will be, yeah, done now. A bonus question. All right.
Well, like many Americans, I get my genetic information from the sports pages. And
Right.
in today's San Jose Mercury News, there's an article about Barry Sanders, who's a running back on the Stanford football team.
Yeah.
And I was surprised to see that
Oh, yeah, the
Stanford
son,
football
right.
team currently has 9 sons of former NFL football players. So as chairman of the Stanford Genetics Department, can you tell us, was Lamarck right?
Well, I don't know. I don't think that argues whether it's Lamarck or genetics. Anyway, I think I couldn't say whether that was genetics or environmental, whether the household may have had the TV on football quite a bit too. That's possible. So yeah, I wouldn't venture into that one. But I do actually think genetics will be very powerful. You actually raise a very interesting topic, because I think there are people out there who have increased bone mass, and you can correlate that with a genetic variant. So there are people who fell out of buildings from 5 stories up who survive, who walk away. Those individuals obviously might be better suited for certain professions than other people, okay? And so I can see where matching genetic information with certain professions, allogotica, does have some value. And I think it's more the opposite, whereas you want to avoid certain things if you're at high propensity for certain kinds of things. So there are changes that are associated with collagen changes with affecting bone and tendons. Those individuals maybe shouldn't be linebackers. So I actually think genetics will impact sports, and it won't be Lamarckian. So, thank you.